Errors in meiosis. By the end of this video, you will be able to explain how nondisjunction leads to disorders in chromosome number and describe how errors in chromosome structure occur through inversions and translocations. Welcome back to Concepts of Biology. We'll continue to read Chapter 7, Section 7.3. Inherited disorders can arise when chromosomes behave abnormally during meiosis. Chromosome disorders can be divided into two categories: abnormalities in chromosome number and chromosome structural rearrangements. Because each small segment of chromosomes can span many genes, chromosomal disorders are characteristically dramatic and often fatal. Disorders in chromosome number. The isolation and microscopic observation of chromosomes forms the basis of cytogenetics and is the primary method by which clinicians detect chromosomal abnormalities in humans. A karyotype is the number and appearance of chromosomes, including their length, banding pattern, and centromere position. To obtain a view of an individual's karyotype, cytologists photograph the chromosomes and then cut and paste each chromosome into a chart, or karyogram. Figure 7.7. This karyogram shows the chromosomes of a female human immune cell during mitosis. Let's look at a career in action. Geneticists use karyograms to identify chromosomal aberrations. The karyotype is a method by which traits characterized by chromosomal abnormalities can be identified from a single cell. To observe an individual's karyotype, a person's cells, like white blood cells, are first collected from a blood sample or other tissue. In the laboratory, the isolated cells are stimulated to begin actively dividing. A chemical is then applied to the cells to arrest mitosis during metaphase. The cells are then fixed to a slide. The geneticist then stains chromosomes with one of several dyes to better visualize the distinct and reproducible banding patterns of each chromosome pair. Following staining, chromosomes are viewed using bright field microscopy. An experienced cytogeneticist can identify each band. In addition to the banding patterns, chromosomes are further identified on the basis of size and centromere location. To obtain the classic depiction of the karyotype in which homologous pairs of chromosomes are aligned in numerical order from longest to shortest, the geneticist obtains a digital image, identifies each chromosome, and manually arranges the chromosomes into this pattern. At its most basic, the karyogram may reveal genetic abnormalities in which an individual has too many or too few chromosomes per cell. Examples of this are Down syndrome, which is identified by a third copy of chromosome 21, and Turner syndrome, which is characterized by the presence of only one X chromosome in women instead of two. Geneticists can also identify large deletions or insertions of DNA. For instance, Jacobson syndrome, which involves distinctive facial features as well as heart and bleeding defects, is identified by a deletion on chromosome 11. Finally, the karyotype can pinpoint translocations, which occur when a segment of genetic material breaks from one chromosome and reattaches to another chromosome or to a different part of the same chromosome. Translocations are implicated in certain cancers, including chronic myelogenous leukemia. By observing a karyogram, geneticists can actually visualize the chromosomal composition of an individual to confirm or predict genetic abnormalities in offspring even before birth. Nondisjunctions, duplications, and deletions. Of all the chromosomal disorders, abnormalities in chromosome number are the most easily identifiable from a karyogram. Disorders of chromosome number include the duplication or loss of entire chromosomes, as well as changes in the number of complete sets of chromosomes. They are caused by nondisjunction, which occurs when pairs of homologous chromosomes or sister chromatids fail to separate during meiosis. The risk of nondisjunction increases with the age of the parents. Nondisjunction can occur during either meiosis 1 or 2 with different results. If homologous chromosomes fail to separate during meiosis 1, the result is 2 gametes that lack that chromosome and 2 gametes with 2 copies of the chromosome. If sister chromatids fail to separate during meiosis 2, the result is 1 gamete that lacks that chromosome, 2 normal gametes with 1 copy of the chromosome, and 1 gamete with 2 copies of the chromosome. Figure 7.8. Following meiosis, each gamete has 1 copy of each chromosome. Nondisjunction occurs when homologous chromosomes, meiosis 1, or sister chromatids during meiosis 2 fail to separate during meiosis. An individual with the appropriate number of chromosomes for their species is called euploid. In humans, euploidy corresponds to 22 pairs of autosomes and 1 pair of sex chromosomes. An individual with an error in chromosome number is described as aneuploid, a term that includes monosomy, loss of 1 chromosome, or trisomy, a gain of an extraneous chromosome. Monosomic human zygotes missing any 1 copy of an autosome invariably fail to develop to birth because they have only 1 copy of essential genes. Most autosomal trisomies also fail to develop to birth. However, duplications of some of the smaller chromosomes, for example 13, 15, 18, 21, or 22, can result in offspring that survive for several weeks to many years. Trisomic individuals suffer from a different type of genetic imbalance, an excess in gene dose. Cell functions are calibrated to the amount of gene product produced by 2 copies or doses of each gene. Adding a 3rd copy or dose disrupts this balance. The most common trisomy is that of chromosome 21, which leads to Down syndrome. Individuals with this inherited disorder have characteristic physical features and developmental delays in growth and cognition. The incidence of Down syndrome is correlated with maternal age, such that older women are more likely to give birth to children with Down syndrome. Figure 7.9. The incidence of having a fetus with trisomy 21 increases dramatically with maternal age. Humans display dramatic deleterious effects with autosomal trisomies and monosomies. Therefore, it may seem counterintuitive that human females and males can function normally despite carrying different numbers of the X chromosome. In part, this occurs because of a process called X inactivation. Early in development, when female mammalian embryos consist of just a few thousand cells, 1 X chromosome in each cell inactivates by condensing into a structure called a Barr body. The genes in the inactive X chromosome are not expressed. The particular X chromosome, maternally or paternally derived, that is inactivated in each cell is random, but once the inactivation occurs, all cells descended from that cell will have the same inactive X chromosome. By this process, females compensate for their double genetic dose of X chromosomes. In so-called tortoiseshell cats, X inactivation is observed as coat color variegation. Females heterozygous for an X-linked coat color gene will express one of 2 different coat colors over different regions of their body, corresponding to whichever X chromosome is inactivated in the embryonic cell progenitor of that region. When you see a tortoiseshell cat, you will know that it has to be a female. Figure 7.10. Embryonic inactivation of one of two different X chromosomes encoding different coat colors gives rise to the tortoiseshell phenotype in cats. In an individual carrying an abnormal number of X chromosomes, cellular mechanisms will inactivate all but one X in each of her cells. As a result, X chromosomal abnormalities are typically associated with mild mental or physical defects as well as sterility. If the X chromosome is absent altogether, the individual will not develop. Several errors in sex chromosome number have been characterized. Individuals with 3 X chromosomes, called triplo-X, appear female but express developmental delays and reduced fertility. The XXY chromosome complement, corresponding to one type of Klinefelter syndrome, corresponds to male individuals with small testes, enlarged breasts, and reduced body hair. The extra X chromosome undergoes inactivation to compensate for the excess genetic dosage. Turner syndrome, characterized as an X0 chromosome complement, as in only a single sex chromosome, corresponds to a female individual with short stature, webbed skin in the neck region, hearing and cardiac impairments, and sterility. An individual with more than the correct number of chromosome sets, 2 for diploid species, is called polyploid. For instance, fertilization of an abnormal diploid egg with a normal haploid sperm would yield a triploid zygote. Polyploid animals are extremely rare, with only a few examples among the flatworms, crustaceans, amphibians, fish, and lizards. Triploid animals are sterile because meiosis cannot proceed normally with an odd number of chromosome sets. In contrast, polyploidy is very common in the plant kingdom, and polyploid plants tend to be larger and more robust than euploids of their species. Chromosome structural rearrangements. Cytologists have characterized numerous structural rearrangements in chromosomes, including partial duplications, deletions, inversions, and translocations. Duplications and deletions often produce offspring that survive but exhibit physical and mental abnormalities. Cri-du-chat, from the French for cry of the cat, is a syndrome associated with nervous system abnormalities and identifiable physical features that result from a deletion of most of the small arm of chromosome 5. Infants with this genotype emit a characteristic high-pitched cry upon which the disorder's name is based. Figure 7.11. This individual with Cri-du-chat syndrome is shown at various ages: A, age 2, B, age 4, C, age 9, and D, age 12. Chromosome inversions and translocations can be identified by observing cells during meiosis because homologous chromosomes with a rearrangement in one of the pair must contort to maintain appropriate gene alignment and pair effectively during prophase 1. A chromosome inversion is the detachment, 180-degree rotation, and reinsertion of part of a chromosome. Unless they disrupt a gene sequence, inversions only change the orientation of genes and are likely to have more mild effects than aneuploid errors. Let's look at change in action, evolutionary change in action. The chromosome 18 inversion. Not all structural rearrangements of chromosomes produce non-viable, impaired, or infertile individuals. In rare instances, such a change can result in the evolution of a new species. In fact, an inversion in chromosome 18 appears to have contributed to the evolution of humans. This inversion is not present in our closest genetic relatives, the chimpanzees. The chromosome 18 inversion is believed to have occurred in early humans following their divergence from a common ancestor with chimpanzees 5 million years ago. Researchers have suggested that a long stretch of DNA was duplicated on chromosome 18 of an ancestor to humans, but that during the duplication it was inserted into the chromosome in reverse orientation. A comparison of human and chimpanzee genes in the region of this inversion indicates that two genes, ROCK1 and USP14, are farther apart on human chromosome 18 than they are on the corresponding chimpanzee chromosome. This suggests that one of the inversion breakpoints occurred between these two genes. Interestingly, humans and chimpanzees express USP14 at distinct levels in specific cell types, including cortical cells and fibroblasts. Perhaps the chromosome 18 inversion in an ancestral human repositioned specific genes and reset their expression levels in a useful way. Because both ROCK1 and USP14 code for enzymes, a change in their expression could alter cellular function. It is not known how this inversion contributed to hominid evolution, but it appears to be a significant factor in the divergence of humans from other primates. A translocation occurs when a segment of a chromosome dissociates and reattaches to a different, non-homologous chromosome. Translocations can be benign or have devastating effects depending on how the positions of genes are altered with respect to regulatory sequences. Notably, specific translocations have been associated with several cancers and with schizophrenia. Reciprocal translocations result from the exchange of chromosome segments between two non-homologous chromosomes such that there is no gain or loss of genetic information. Figure 7.12. An inversion occurs when a chromosome segment breaks from the chromosome, reversing its orientation, and then reattaches in the original position. A reciprocal translocation occurs between two non-homologous chromosomes and does not cause any genetic information to be lost or duplicated. This ends Chapter 7, Section 7.3, Errors in Meiosis. In our next video, we'll review key terms and summarize Chapter 7.